Alright, so no analysis because I don't know how to do Bayesian analysis. But what I thought I would do is tell you about a problem which is biological and real biological data has been collected in the sense that it's much more laboratory oriented data. We haven't collected the data, so it's a collaborative work with Joshi from JCA. So I'm going to do two things. First of all, I'm going to, since I'm lucky that there are people here, many people are here from physics, so I will take that opportunity to set up the model, which is a differential equation based model, which is based on biological phenomena that underlies the process. And second, I will just show you a little bit of very simple analysis of data that we got from collaborator. And then I will leave I would like to discuss with all of you, those who are interested later, that how can I actually use this model and this data to parameterize the model, because it has about 7 parameters, and set up a Bayesian analysis model. I have a collaboration with another person, Tiata Camp, where they are trying to set up a Monte Carlo Markov chain method using the inverse problem theory, and trying to do Bayesian, but I don't know, so I would like I would like to take this opportunity to learn that. Okay, so when I talk about, as you can see, that this is an area which is slightly more of organismal biology, where we are looking at how do the population of an insect called Drosophila increase or decrease, and how do we analyze. So when we talk about population, actually it can be anything. It can be any unit which is capable of growth, and we can measure it in an isolated patch. So it becomes a single population. When we measure it over ecological area, then it becomes interacting population. What determines? Essentially growth, what we call as birth and death. There are other ecological phenomena that contribute into the distribution in a body, but essentially net growth decide what is going to be the survival dynamics and what is going to be the extinction. And the reason that we do this kind of a study, or people do, ecology is one of the oldest area where mathematics has been applied, statistics has been applied to study models and data, because we want to predict based on what we have or what we observe, we would like to predict whether a particular population is going to grow, if it's a rare species or if it's a pathogen population. A disease-causing agent, and we would like to know whether this disease is going to grow in the population or not. So that's why it's very important to study these things. Now, before I go into showing you what we do, there are two types of growth that people talk about in ecology. One is continuous growth, with which we are more we know more about it, because we all do that. You have grandchildren, parents, and grandparents coexisting, which is called continuous growth, where generations overlap. But there are a lot of organisms, including the insect I'm going to talk about, their generations don't overlap. After laying the eggs, the adult dies. So the next generation actually never sees the older generation. And there are other organisms where there is a structure. At a particular age, they reproduce at a particular stage. So they can also be modeled using this, they are called discrete growth systems. And in mathematical terms, these sorts of discrete growth systems are modeled using discrete equations, and continuous growth systems are modeled using continuous differential equations. Let me just show you that even though in the simplest possible model that people study in differential equation, the single species growth, all it does is it just stabilizes, population growth stabilizes to a particular point. But if you are studying the simplest possible discrete growth equation, which every physicist probably has some point of time studied the logistic equation, depending on the parameter of growth rate, it can show many different types of dynamics. All this is fine, because what people did was physicists used these kind of equations which model single population. These are all one-dimensional maps, and they showed that these one-dimensional maps have a very general and universal kind of dynamics. As you increase the growth rate, they go on to show period doubling bifurcation, and then period doubling bifurcation, which leads to chaos. and therefore they put all these models into the same universality class. And the effect of that was on ecology is that people use these models interchangeably when they were trying to model data. A long time back, we did some amount of work and we showed that actually the universality breaks down among these models when you use small perturbations, and these are perturbation strength, and I have just shown two examples of two types of maps which are shown here. One is this and one is this one. So you can see the nature of the hump here, and we showed that under small perturbations actually they show very different types of dynamics, because this is when there is no perturbation. For positive small perturbation, this map actually goes into chaos, whereas this map actually stabilizes. And we did some more analysis and showed that the maps of tails or inflection points show a very different behavior compared to the other one. So what we want to get from this understanding is the nature of density dependent, which essentially decides this particular, how does population respond when the density is higher. So crowd, response to crowd, actually decides what is going to be their dynamics under small perturbation. These perturbations could be ecologically important features like immigration and migration, which happens in a lot of situations. So the question is, these are single species models. They fit experimental population recursion data to equations using regression techniques, and the only criteria for choosing a model is goodness of fit. But they actually do not provide any detailed explanations of regulatory mechanisms that are occurring at very different life stages of organisms. I don't know how many of you have done these experiments when you were a kid in school, that you catch hold of a caterpillar, put it in a shoebox with some leaves, and then allow it to grow, become a butterfly. We did it because in our school we always had a science class and lower classes outside the classroom. So we know that it starts from an egg, it becomes a larvae, which is a caterpillar kind of a state. Then it goes into a shell, which is called the pupa, and then that shell, inside the pupa, a lot of developmental mechanisms occur, and then the shell breaks out and the adult emerges as a butterfly. And this butterfly or this insect then, on being adult, lays a number of eggs and dies, and then the next generation starts. So there are a lot of different, there are these different life stages of organisms, and there are several regulatory mechanisms that occur. So how do all these regulatory mechanisms operate? Operating at different stages combine to produce one or two parameters, a single model still remains unresolved, and essentially those models are not useful. So what we wanted to look at is adapt these simple models to more complex models that consider life history stages and test it in laboratory populations of these particular kind of insect, which is very popular with biologists. So a very short account of this is the insect, which you see if you leave a banana outside, they will all have these small black insects flying around. That's a fruit fly, and it has several distinct life stages. As I said, there's egg, then there's several different stages of the larvae, and then it becomes the pupae, and then all the development processes occur, and then it becomes adult. It lays eggs, and this one dies. So all these different stages actually show very different, very many different kinds of density-dependent processes. So if I want to predict that given the number of eggs, I want to know how many number of adults I'm getting, because that is the prediction I would like to make. That with the number of adults at this generation, can I predict the number of adults at the next generation? Then what I'm doing is I'm integrating understanding of reproduction, growth, and mortality at the individual level to density-dependent regulation at the population level processes. So this interaction becomes very important in this kind of stage-specific systems. So here is you can forget about how it looks, and you can just concentrate on this kind of a schematic where after the egg is laid, then the egg becomes larvae. They have to be viable, that is, they have to live to become adult, and then these adults will lay eggs again, and that becomes the next generation. So this is how the process goes on. And if you look at the larval state, so let's look at this state, because this is one of the in this case, there are several different factors that regulate whether this particular individual larva is going to become adult. Survival is of course very important. Feeding rate, foraging behavior, response to crowding, that are very important. Survival and fertility rates of adults also affect the levels of crowding and nutrition of the larva. Okay. And the adult state, these are these loops which occur. It is determined by adult size, adult density, adult nutrition. So all these factors actually decide that how many eggs this is going to lay. So it is not a simple model that people have been looking at. So how do people go about developing a complex model? Of course, looking at experimental data. In ecology, a lot of people have been very mathematically oriented. So they did some experiments by increasing, they started cultures with different numbers of adults, and they wanted to know different numbers of eggs, and they wanted to know how many adults come out of these eggs after going through the larval stage. And they did plots like this. So this is number of adults coming out based on number of eggs. This is number surviving out of the initial egg density. And this is then a proportion of surviving egg to adult from egg density. So based on all these kinds of experimental results, what they did was they fit those, sorry, I don't want to talk about this. Based on all these experiments, what they did was they developed these expressions that how does egg density decide larval survivorship? How does egg density decide adult quantity? Or how do they depend on each other, not really decide? How does adult mean weight decide egg density? This is a kind of indicative of nutrition. And how does adult density decide how many adults are going to, how many eggs it's going to lay? So all these are experimental data. Based on this, what they did is they fit functional forms, and these functional forms, they had some parameters which they, based on the data they fitted. These are simple fitting of functional forms, and what we have done is we have taken these functional forms with so many different parameters that one can look at, and here are the 4 functional forms that we need to use now and make a model out of it. Now I'll just list out the parameters that are coming here. The problem with this, why is it that we can't use these as a model to predict what is going to be the next number of adults in the next generation based on number of adults in the earlier generation? For the simple reason, because in experiments you can never measure the number of eggs, you can never measure the number of larvae, but the way the experiments are done, they all go into these very mushy kind of agar, and getting them out and measuring them is just beyond none of the experimental people would like to do it. So what we did was, we did a very simple thing. We took these 4 expressions and we just got small nt, which is actually the number of eggs equivalent to number of larvae, out of that, out of these 4 expressions, and that became our, I'll come back to this. This is the so-called model, which depends not only on capital N, which is the adult, and the weight of the adult. So what we have done now is, earlier it was a single variable model. Now we had to include 2 variables because of the stage specificity given in its life cycle. But we have been able to get how does nt plus 1 depend on nt or wt, and how does wt plus 1 increase at the next time step on the earlier number of adults and the earlier weight. So both are measurable in the laboratory and field. And this is the kind of set of parameters that these papers give. These parameters are highly dependent on the species of insect Drosophila you are working with, or also on the labs.
Another question.
Yeah.
So is the problem given nt and wt to infer all the parameters which occur in the model? Is that the...
So that's one problem. That's one of the problems. So given that all those 7 parameters, they have some numbers, but they don't match when you're trying to do this experiment because they change based on the different species you are using. So what I'm going to show you here is use those parameter values, and if you look at this model and try to understand what is the dynamics that this model is capable of showing, because finally we are going to, in lab also we are going to study the time series of how many numbers at what time. So this is the kind of diagram, these are bifurcation diagrams for those who understand. So these are actually the values that I showed you just now, which shows that the entire system that we are looking at is actually in a fairly complex chaotic state. But if I can change some of these parameters, then I can take the system onto more regular state. This is what the model says. Now we don't know whether this model does not consider data at all, except setting up of these functions. But this model does not tell you that actually when you do experiment, this is the end you will get for all time, for all the time. So that is the time we looked at the data that was given to us. The data was simple. So this is how they collect data. You don't have to worry about this. Basically, what they do is they start with a pair of males and females insects, and they keep them in a particular glass vial with certain amount of food. They give it to the larvae and the adult. Those are the two stages where they can eat and move. And those, so they start with a certain number of adults. They lay eggs, then they become larvae. They eat the food, and they give rise to a number of adults again. And those adults are taken away. So this is essentially discretizing the generation, because you don't allow them to stay together. You can, since there are two stages where they eat, you can actually change the level of food in these two stages to find out what is the role of nutrition on the population dynamics of this the adults. So what do you get? What kind of so as you can see, the number, one of the parameters that one takes actually depends a lot on which lab you are working on, because even if they are all Drosophila, a particular species, but they still, because of the way they are grown, they have very different parameter values. This one value that I showed you. So how is the data set? Let's look at the data set. Okay. So here are 4 sets of data. Each set has 8 replicates. So it's one of the best kind of data that one could take. There are 50 generations plotted here. This 50 generation took about 2.5 to 3 years to generate. So actually when we gave this project to mathematical sciences and DST, they said come back with more data. So I went to my collaborator and he said tell him that my grandchild will come back with more data. Because it's just not possible to generate a very large set of data, and that is another problem, that why we need a whole lot of statistical analysis of this, on these kinds of problems. Okay, so what are these 4 sets? Each set has 8 replicates. So what the first position and second position? I told you that we can give certain amount of food to the larvae and certain amount of food to the adult. And what the first position is for food to the larvae, second position is for food to the adult. And here is low food to larvae, high food to adult. High food to larvae, low food to adult. And both the stages getting low food, both the stages getting high food. The aim of this kind of work is to show how flies, because people want to understand how nutrition affects population dynamics, how flies adapt to different types of food regime. And you can see the scales are all the same. You're plotting the number of adults. This is number of generations. And you can see, being the scales being the same, you can see that this is one type, this is another. A lot of noise is there because you can see that the fluctuations are much kind of larger amplitude here. It seems more regular but smaller amplitude, etc., etc. And all these are values which you need not worry about. Question is, does this data set, how do I say that this data set actually, my model that we have developed would be able to validate this data set or would be able to simulate this data set?
So in all cases it is regularly going up, down, almost regularly.
Most discrete equations do that, and the reason for that is that whenever there's a very high, it's understandable also, whenever there's a high population, actually high population means what? Large number of adults. So large number of adults would normally give rise to large number of eggs. But when there's large number of eggs, large number of larvae, they compete with each other. So that competition kills a large number of them because there's only a certain amount of food. So essentially there's a crash at the population size. Lower number of adults, if they're healthy, if they get good food, they lay larger number of eggs, and then the procedure goes on. So that can be, we did want to see what is the role or what is the effect of these nutrition in these two-stage insects can have on the population dynamics. And this is what it is. Now, how do we know that this is? Yeah.
So sometimes it may happen that when the population size becomes very low, so that they may not be able to.
Right. So they just you have to restart the whole experiment again, and it happens quite regularly. So in a different way of looking, if you plot the mean weight and population size at 2 consecutive generations, this actually gives you a much better idea about how these different food regimes look when you plot them in this. And these two seem to be more structured, and these two actually should show me more periodic oscillatory event. That this should show me a more stable fixed point behavior. But look at the amount of noise that you see there. This is the data, and this is the data we need to use to parameterize the model. And I told you how the model was developed. What we have done is a slightly different way of looking at it. The same lab actually did work with the same organism at 3 different PhD students at 3 different non-overlapping time. And what we have plotted here is you take the population size, you make a histogram, and you just plot the histogram. Okay? So you can see that one of the students had a data set of 49 generation, 8 replicates, which I am talking about. Somebody else did a 32 replicate system, but only 11 generations. Somebody else did a 36 replicate with 21 generations. And if you make histograms of the size distribution, they more or less look similar. So one thing is clear, that as long as the food regimes are kept similar, the data set shows quite similar structure. If you take the model that we have built and you do simulation of models, and then we follow this one. So we start with 8 replicates. We start with 8 pairs. We also started with 8, which means the initial condition was 8, and we kept doing that, and we got something which looks similar. Though there are differences there. The most interesting thing that comes out from this data set is, so you have this bimodal distribution. This is experimental data. When you do this data and simply look at the data, you have the population size has 2 modes. This is also the mean weight. If you do the distribution, that also has 2 modes. So simply one would think that the ones with the high population, this is high population density. So lower population size actually should show low weight, so essentially I am trying to say that there could be a 1:1 correspondence between the weight and the population size. But that is not what it shows. If you look at this, if you take since I have the data set, if you take the low population size data set, go back and look at what was their weight at the t-1, actually it shows both it has some adults which are which are very big and thick and fat, and most of the adults are thin. And if you go and look at this one, the second one, and you can see that it had a more distribution. So this had to be described. It was not very difficult to describe. You can look at this, the numbers also, if you go backwards, because you have the data set and you can see that this is A and this is B mode. So A mode actually has a distribution size which is far wider, whereas the B has a more stricter distribution which is coming from a smaller size. This essentially means, these 2 pictures, is that you can have this, and that's the slide I actually skipped because it takes a little while, and that was a major problem in ecology, that it says that you cannot use single species model in that because there's a degeneracy there. You can get the same population size from 2 different population sizes. Okay? So it is impossible to go from initial value to the next one, because that's where the difference is, because this is n-1 and this is n. So you actually, it's quite difficult. Both of them have different weights, but they are having the same number. So this is a problem which has always plagued population ecologists, that in these kinds of models, I cannot start with the initial value and go to the next value because there's degeneracy here. So that is why what we have proposed is you need one more.
Accidental or is it generic?
Excuse me?
This kind of degeneracy, is it accidental to some particular setup? Or?
No, that is why I showed that all of them show same results. So if you go and work with other sets of data, you see the same. And it's understandable if you consider why it's happening, because what is happening is you can get the same number, but the insects can be actually large and fat. But you can get the same number when the insects are small and thin, and that is how we had to bring in the second parameter, which otherwise people can't bring in, and that is what resolves. So when we do the degeneracy problem, when we study the degeneracy problem, we had to separate this state space into multiple. Given that, you can see if you can use a model also generates synthetic time series from there, and you can show that they also show the same kind of this peak 1 shows two different types of sizes, peak 2 shows this one, and here also theoretically also this feature at least is very well simulated in the model. So we have some reasons to believe you can continue to do that, and of course when you look at the time series they don't match very well, but you can these kinds of structures, it's a highly noisy experimental time series. We can take the minimum, this is a deterministic model, so we can add a little bit of noise in the minimum, and we can convert, one can convert this to look at it like this, but again everything is parameter dependent. So it is, I don't think I have much to say beyond this. But the problem is that the problem that we are facing or we are wanting to do is, can we use these 4 into 8, that is 32 sets of time series data, to build a model which will not be, we have a model, we have a set of data. So essentially we are trying to use both to arrive at a set of parameters which should give us a match. And given the kind of phenomena that are involved here, there's so much of noise that is involved, because these are all whole organism phenomena. It's most likely that we would require a much more detailed analysis using some priors. What exactly are the priors here? What are the conditions here? What kind of conditionings are involved? What are the features? Because it's not only it's food, it's the earlier weight, it's the earlier number, several different points are involved. So it would be nice to actually be able to physically write down, because since I'm not a mathematician, I come partly from physics, I would like to know the process rather than just expressions, mathematical expressions. Then what are the points that could physically and biologically be important to consider as prior, that we should know, take as prior information, which parts of the data and which parts of the information. I would stop here because, as you see, there are no Bayesian analysis here. This is the person who did the modeling part. These are the people who generated the data. All the work was done here, so I'll just move forward. Thank you very much.
That the growth genomics of Drosophila is a discrete generation event.
It's not discrete generation event, but in the experimental setup they have made it discrete by taking the adults away after they have laid eggs. So you take it away, then it becomes discrete.
Other questions? Okay, if not, let's thank Chandra.